see them at all, you have to use a microscope. Most of the specimens that are studied with a microscope like this, a light microscope, have been fixed and embedded in wax. But using suitable optics, it is possible to look at living cells in a light microscope. These are fibroblasts, connective tissue cells which secrete collagen. They've been magnified several thousand times and filmed with time-lapse photography so that you can see them moving. The most obvious thing inside the cells is the nucleus, with its dark edge. If you look carefully, though, you may also be able to see some thread-like structures in the cytoplasm. These are the mitochondria. They are only a few micrometers long, and it's difficult to make out any details of their structure. You might think it would help if the magnification was increased. All right, have a look at a section of liver. Here it's magnified about 500 times, and it's stained with eosin and hematoxylin to improve the contrast of the image. At this magnification, the image appears crisp, but the individual cells are too small to be seen clearly. You can see the nuclei, they're the dark blobs, but not much else. If the magnification is increased, so that the cells are enlarged to about 1,000 times, the image gets bigger, but you can't make out any more details. This is because the resolution of the microscope is limited to about half the wavelength of the light used. With visible light, this means that the resolution of the light microscope is limited to about 1/5 of a micrometer. So any particle which is smaller than this won't be seen at all. And any two structures which are closer together than 1/5 of a micrometer won't appear as separate structures. The only way to improve the resolution is to use a much shorter wavelength. This can't be done with a beam of light, but it can be done with a beam of electrons, which is what happens in an electron microscope. Moving electrons show wave properties, and the faster they move, the shorter their wavelength. In the electron microscope, electrons are accelerated by a very high voltage, which gives them a wavelength much shorter than that of visible light. In practice, this means that the resolution in the electron microscope is about 100 times better than in a light microscope. Have another look at the section of liver in the light microscope when it's magnified as much as possible. And now look at the section in the electron microscope and see how much clearer the nucleus and mitochondria are. And this is at the same magnification, about 2,000 times. As the magnification is increased, more and more details of the nucleus and mitochondria appear, and you can see structures that you couldn't see at all in the light microscope. The endoplasmic reticulum with its tiny ribosomes. The Golgi body. The lysosomes, they're the dark structures. And the plasma membrane on the left, the outer membrane of the cell, which keeps the cell intact and separates it from its neighbours. Using the improved resolution of the electron microscope, which not only gives clearer pictures at low magnification, but allows the magnification to be increased considerably before the clarity is lost, a whole new world is revealed. Bacteria, which are about the same size as mitochondria, can be seen in detail. Viruses, about 200 times smaller than most cells, can be seen clearly. And it's even possible to see individual molecules. The fine threads on here are molecules of DNA released from a damaged bacterial virus. These photographs, or electron micrographs, were all taken using a transmission electron microscope. In this type of microscope, negatively charged electrons are accelerated by a very high voltage from a heated filament at the top of the column, which acts as a cathode, to a positively charged anode. The stream of electrons emerging from the anode is focused into a beam by a powerful electromagnet called a condenser lens. The electron beam then passes through the specimen and is finally focused and enlarged by a system of electromagnets, the objective, the intermediate and the projector lens, to give an image on a fluorescent screen, like the image on a cathode ray tube of a television screen. The light and dark areas on the screen are related to the dense and less dense areas of the specimen. In an electron-dense area of the specimen, some of the electrons are scattered, so the electron beam arriving at the screen is less intense and the image appears darker. In a less dense area of the specimen, most of the electrons will pass through, so the electron beam arriving at the screen is more intense and the image appears lighter. The electron density of the specimen depends on the atomic number of the atoms that are present. The higher the atomic number, the more the electrons are scattered, and the greater the contrast of the image. Because most of the atoms in biological materials have a very low atomic number, carbon, hydrogen, oxygen, for example, they don't scatter electrons well and don't produce an image with much contrast. To improve the contrast and get an image like this, the specimen is treated with the salt of a heavy metal like uranium or lead. Where the metal stains the specimen, the electron density in that area is increased, producing a much greater contrast in the final image. Using controls at the side of the microscope, the specimen can be moved around in the electron beam so that you can see different areas of it. And if you want to take a photograph of what you see, the fluorescent screen is moved out of the way and the electron beam focused onto a photographic plate or film which is also sensitive to electrons. The whole system, from the heated filament at the top of the column where the electron beam starts to the screen where the final image is seen, is kept under vacuum, so the electron beam isn't interrupted by air molecules. The specimen in its holder must be put into the microscope through an airlock, and it must be specially prepared. The electron microscope can't be used to study living cells because they can't withstand the high vacuum. The specimens must also be extremely thin so that the electron beam can pass through them. This means that either the specimens must be very small, like viruses or protein molecules, or they must be cut into thin sections, 1,000 times thinner than a piece of writing paper. So that they can be put into the microscope, specimens are mounted on copper or nickel grids, which are only 3 millimeters in diameter. Each grid is made up of a meshwork of open squares. The specimen is supported by the metal lattice, while electrons pass through the holes in between. For some specimens, a suspension of viruses for example, the electron density is increased in one of two ways to produce a high contrast image like this. The first, called shadowing, involves coating the specimen with a heavy metal. The specimen is put into a vacuum chamber and then subjected to a beam of heavy metal molecules evaporated from a heated filament. The metal condenses around the specimen, and when viewed in the electron microscope, it casts a shadow, which makes the specimen stand out more clearly. Like these collagen fibrils from human skin, magnified about 100,000 times. The alternative approach, known as negative staining, involves surrounding the specimen with a solution of a salt of a heavy metal, uranium for example. When the salt dries, it forms an electron-dense layer around the specimen. So the specimen appears light against the dark background. This technique is particularly useful for seeing the surface features on small specimens. These are fragments from the inner membranes of mitochondria. The small knobs sticking out from the surface are formed by the enzyme complexes involved in oxidative phosphorylation. If the specimen is too large to be studied whole, it must be sliced into thin sections. First, though, the structure of the specimen is stabilized by treatment with chemicals, glutaraldehyde and osmium tetroxide, so that as much detail as possible is preserved when the specimen is cut into sections. The specimen is then dehydrated by soaking it in alcohol solutions of increasing strength, and finally it's embedded not in wax but in a block of epoxy resin. It's from a block like this, only a few millimeters across, that the thin sections are cut, using a knife made of glass or diamond. The sections come off onto water and are picked up on one of the special support grids. They can then be stained with heavy metal before being put into the microscope. Usually a number of sections are cut from one block. You can see them coming off the knife like a ribbon. Each section contains a slice of the specimen taken at a slightly different level. So, by putting the images obtained from each section together, it's possible to build up a three-dimensional picture which shows the shape of the specimen itself or the shape of structures within it. Because the processes involved in preparing a specimen for electron microscopy are very harsh, there's always a risk that some alteration of structure will take place or that artifacts will be introduced. To minimize this risk, the techniques of freeze fracture and freeze etching have been introduced. In the freeze fracture technique, the specimen is rapidly frozen to the temperature of liquid nitrogen, that's minus 196 degrees Celsius, or even lower. The water in the specimen freezes, which holds everything in place, and the frozen specimen is then put into a vacuum chamber and split with a knife. The specimen always fractures through the line of least resistance, and the fractured face looks like a lunar landscape. If you look at this electron micrograph of the nucleus of a liver cell, you can see the nuclear pores. In the case of freeze etching, the specimen is also frozen, but the temperature is then raised slightly to allow a thin layer of ice to be removed by sublimation. Part of the specimen then stands proud of the surrounding ice. This is shadowed with a layer of heavy metal, and then has a strengthening layer of carbon added to it. The specimen is dissolved away with sodium hydroxide, and the carbon replica is recovered on a copper grid and examined in the microscope. This technique gives quite distinctive results, as in this micrograph of a piece of placenta. Most biological materials can be examined in the electron microscope using one or more of these techniques. Because more structural detail is being revealed all the time, more is being learnt about the form of living things and the relation of form to function, whether in man, microorganisms, or molecules. In every living cell, thousands of chemical reactions are taking place all the time, building up, breaking down or changing molecules to provide the energy and the materials the cell needs in order to function properly. Cells are so small that their structure can only be seen in any detail in an electron microscope. But even magnifying a cell many thousands of times doesn't reveal its molecular organization, and in any case, you can't see the chemical reactions going on inside it. This electron micrograph of a liver cell, for example, shows that the nucleus is big and has a certain shape, but it doesn't show the double helix of the DNA. It shows that the mitochondria are small and elongated, but it doesn't show the enzymes of the citric acid cycle. To find out what each part of the cell does, rather than what it looks like, the cell has to be taken apart and analyzed chemically. Isolating the different parts of the cell from one another involves a procedure called subcellular fractionation. In principle, what happens is that cells are broken open so that their organelles are released. The resulting mixture of nuclei, mitochondria, lysosomes, ribosomes, and cytoplasm is then centrifuged at different speeds using a very high-speed centrifuge, usually called an ultracentrifuge. Each type of organelle is separated from everything else in the mixture, producing a series of subcellular fractions, hence the name. In practice, the separation isn't nearly as clear-cut as this. In the ultracentrifuge, organelles are separated from each other on the basis of their size and density, and some organelles are very much easier to isolate than others. Have a closer look at the liver cell and you'll begin to see the problem. Apart from the obvious structures, like the nucleus and the mitochondria, the cytoplasm contains lysosomes and a number of even smaller organelles, like peroxisomes and ribosomes, which can't be identified very easily on this micrograph. The cytoplasm also contains the complex network of membranes known as the endoplasmic reticulum, and the system of flattened sacs and vesicles called the Golgi apparatus. And around the outside of the cell, there's the plasma membrane. When the liver cell is disrupted before ultracentrifugation, these delicate membrane systems break into small fragments, which then seal themselves to form tiny vesicles. This means that not only is it impossible to isolate the endoplasmic reticulum, the Golgi apparatus, and the plasma membrane in an intact state, but also the mixture of tiny cell components to be separated by centrifuging is even greater than it is in the cell, with many of the components having a similar size and density. In general, the easier it is to isolate an organelle from the cell, the more that organelle has been studied, and the more information has been obtained about its function. Using suitable conditions, mitochondria can be isolated quite easily from liver cells by the process of subcellular fractionation. First, the liver is chopped into pieces using a pair of scissors. A buffer solution is added. And the liver cells are then broken up using a piece of apparatus called a homogenizer. When it's switched on, the plunger rotates at high speed, and as the glass vessel is moved up and down, the cells of the chopped liver are forced through the narrow gap between the plunger and vessel wall. This disrupts the cells and produces a homogeneous mass of cell pieces. Finding out what are the least damaging conditions for disrupting the cells and releasing organelles is an important part of research into this technique. The right conditions can only be found by trial and error. If the mitochondria are to remain active during the isolation procedure, so that they'll be suitable for biochemical studies, various precautions have to be taken. The temperature is kept low with ice to protect the mitochondrial enzymes and prevent bacterial growth, and the solution which is added to the chopped liver before homogenization is buffered to prevent changes in pH. The solution also contains a large concentration of a metabolically inert substance, in this case sucrose. This is so that the solution will exert an osmotic pressure roughly equal to that inside the mitochondria, and so prevent the organelles from bursting as they are released from the cell. To separate the mitochondria from this homogenate, it must be spun in an ultracentrifuge. The homogenate is poured into thick-walled tubes, which are put into a metal rotor and spun in a refrigerated chamber. The speed and length of rotation are set by these controls. If the homogenate is spun at a low speed, so that the centrifugal force developed in each tube is about 1,000 times the Earth's gravitational force, the nuclei are pulled towards the bottom of the tube. Being the biggest organelles, they're the first to separate out. After 5 to 10 minutes, the nuclei are packed down into a pellet at the bottom of the tube, with all the other organelles still in suspension in the supernatant. The supernatant can be poured off into another centrifuge tube, leaving the pellet of nuclei behind. At this stage, the mitochondria, which are smaller than nuclei, are still in the supernatant. To pull them down to the bottom of the tube, the centrifuge speed must be increased to develop a force of about 3,000 times gravity, and after 10 minutes, there's a pellet of mitochondria. The supernatant now contains all the cell fragments that are smaller than both the nuclei and the mitochondria, and it can be centrifuged again and again at progressively higher speeds to pull down other organelles and complete the cell fractionation. So, starting with the tissue homogenate, first the nuclei are pulled down, then the mitochondria, then the lysosomes, and finally the cell membranes. The pellet formed by the mitochondria is cream-coloured and gelatinous, and almost certainly it won't be completely pure. The mitochondria will be contaminated by other organelles like lysosomes or peroxisomes. Big lysosomes, for instance, they're the dark structures, are not very different in size from the small mitochondria below them. To purify the mitochondria, the pellet is first resuspended in liquid. Some of the suspension is then put on top of a buffered sucrose solution in a centrifuge tube. The solution in this case is very special. It's got a high concentration of sucrose in it, so it's a very dense solution, but the really important thing about it is that the tube is set up in such a way that the density of the sucrose solution increases as you go down the tube. In other words, there's a density gradient. When the tube is centrifuged at an appropriate speed, the mitochondria and any other organelles in the suspension at the top of the tube will be pulled towards the bottom. But when they reach a part of the tube where the density of the sucrose solution is the same as their own density, they'll stop. Because mitochondria, lysosomes and peroxisomes, for example, all have different densities, they'll stop at different places, forming a number of separate layers or bands, and each of these can be collected separately for analysis. This is the cream-coloured mitochondrial band, which should now contain mitochondria uncontaminated by other organelles. The most obvious way to check on the purity of this fraction is to look at it under the electron microscope. Here's an electron micrograph of mitochondria prepared in the way you've just seen. It shows that the mitochondrial fraction is fairly pure. There are only a few organelles present that are not mitochondria, and the mitochondria look more or less the same as they do in the whole cell. In other words, they haven't suffered any gross physical damage or disruption during the isolation procedure. But these things alone don't show that the mitochondria are still biochemically intact and active. A better check on their state is to see whether they contain the right enzymes in the right amounts, and whether they can carry out a normal mitochondrial function like respiration. In mitochondria there are certain enzymes, like the enzymes of the citric acid cycle and those of the respiratory chain, which are not found in any other parts of the cell. Any of these enzymes can serve as an identification mark for mitochondria while they're being isolated, and one that's often used is succinic dehydrogenase, an enzyme of the citric acid cycle. If the activity of this enzyme is measured in a liver cell homogenate, and then in each of the subcellular fractions obtained from it by centrifugation, the nuclei, the mitochondria, and other fractions, it's possible to follow the progress of the mitochondria as they are separated from other cell components. Ideally, if all the mitochondria have remained intact and have been recovered in the mitochondrial fraction, the activity of the enzyme in the liver cell homogenate should be the same as the activity recovered in that fraction. In practice, there are always losses, but the amount of enzyme which is recovered in the mitochondrial fraction gives a good indication of the biochemical state of the isolated mitochondria. A preparation of mitochondria which looks good under the electron microscope, has all the right enzymes, and will take up oxygen when given an oxidizable substrate, can be used for further studies. By isolating cell organelles and then studying what they can do, biochemists have gradually been able to discover which organelles are responsible for which chemical reactions and processes in the cell. The energy-producing organelle of eukaryotic cells is the mitochondrion. These are mitochondria, magnified 10,000 times in an electron microscope. They're small, rounded organelles, and they're about the same size as bacteria. They're surrounded by two membranes, an outer one which is fairly smooth, and an inner one which is highly folded. It's on this inner membrane that energy production takes place. Energy used by the cell is chemical energy in the form of adenosine triphosphate, or ATP. So energy production in the cell means ATP production. Mitochondria produce ATP by taking partially broken down nutrients from the surrounding cytoplasm, sugars, fats and amino acids, and oxidizing them to carbon dioxide and water. The whole oxidation process, often called respiration, involves two series of enzyme-catalyzed reactions: the tricarboxylic or citric acid cycle and the electron transport chain. You'll probably study the details of these in another part of your course, but briefly what happens is this. During the reactions of the citric acid cycle, electrons are released from nutrient molecules. They then flow along the electron transport chain from one electron carrier to another, and at the end of the chain they combine with oxygen and with hydrogen ions to form water. Energy is released as the electrons pass along the chain to oxygen, and this energy is used to drive a reaction in which a phosphate group is added to ADP, forming ATP. This way of forming ATP is called oxidative phosphorylation, as the overall process involves the oxidation of nutrients and the phosphorylation of ADP. It only happens when electrons flow along the transport chain. The process of oxidative phosphorylation can be studied in a laboratory using mitochondria which have been separated from all the other cell components. Mitochondria are isolated from cells by breaking the cells open and then centrifuging the contents at different speeds. That's the way the mitochondria in the bottom of this tube have been prepared from liver cells. At the moment, the mitochondria are packed down at the bottom of the tube, but by adding a suitable solution and stirring gently, they can be dispersed into a suspension. And using a suspension like this, it's possible to measure both the uptake of oxygen and the production of ATP by mitochondria. Oxygen uptake is measured using an instrument called an oxygen electrode. This is a device which measures the concentration of oxygen in a solution. It consists of two pieces of metal, one silver and one platinum, which are immersed in an electrolyte, saturated potassium chloride solution. A piece of lens tissue is put on to act as a spacer, and then a small piece of plastic film to keep the potassium chloride in place. The two pieces of metal are connected through the black lead to an electrical circuit, which applies a small voltage across them, and makes the platinum act as a cathode and the silver as an anode. When a solution containing oxygen is added to the electrode chamber, oxygen from the solution diffuses through the plastic film and becomes reduced at the cathode. The cathode is depolarized, and this causes a small current to flow from the electrode. After being amplified, the current is recorded on a chart recorder. How much current flows depends on how much oxygen reaches the cathode, which in turn depends on the oxygen concentration of the solution added to the electrode chamber. The greater the concentration of oxygen in the solution, the larger the current. The chart recorder is usually calibrated so that one side of the chart represents the current flowing from the electrode when no oxygen is present, 0% oxygen concentration, and the other side represents the current flowing when the oxygen concentration is 100%. 100% here means that the solution is saturated with oxygen at the temperature being used for the measurement. Between 0% and 100%, the scale is linear, so it's easy to calculate the oxygen concentration at any intermediate point if you know the solubility of oxygen in water. During an experiment, the chart paper moves forward at a constant speed, so it records not only the oxygen concentration at any moment, but the change in oxygen concentration over a period of time. In other words, the rate of oxygen uptake. To measure the rate of oxygen uptake by the isolated mitochondria, a few milliliters of the suspension are put into the electrode chamber, which is kept at 37 degrees Celsius, body temperature, by a water jacket supplied from a heated water bath. A tiny magnetic stirrer is dropped into the chamber as well to keep the suspension of mitochondria stirred. This makes sure that the oxygen concentration is kept the same throughout the suspension. The chamber has a close-fitting lid to keep out atmospheric oxygen, but there is one tiny hole in the lid. so that other substances can be added to the suspension if required. If you remember, to carry out oxidative phosphorylation, mitochondria must have a nutrient substance to oxidize, a supply of oxygen, some ADP, and some inorganic phosphate. Of these, oxygen is already present, dissolved in the liquid in which the mitochondria are suspended. And inorganic phosphate is present because the suspending liquid is a phosphate buffer. That leaves nutrient and ADP. When a nutrient is added to the mitochondria, in this case pyruvate, a product of sugar breakdown, nothing happens on the chart recorder. And this is because there's no ADP present. The amount of ADP in mitochondria is always very small, and when mitochondria are given an oxidizable substrate and some oxygen, they turn all their ADP into ATP, and then they stop respiring. And they won't start again until they're given some more ADP. Have a look and see what happens when ADP is added. There's an almost instantaneous uptake of oxygen as the added ADP is combined with inorganic phosphate to form ATP. And when all the ADP has been converted to ATP, respiration stops. Here's another demonstration. ADP is added, and oxygen uptake increases until all the ADP has been turned into ATP. This shows that oxidative phosphorylation is a very tightly controlled process. It only takes place when there's a need for ATP, and the signal that ATP is required is an increase in the ADP concentration. Think about muscle cells. They get their energy for contraction by hydrolyzing ATP to ADP. So, as the muscle contracts, the ATP concentration falls and the ADP concentration rises. The muscle will only continue to contract if the ATP supply is replenished, and it's the rise in the concentration of ADP which signals to the mitochondria that more ATP is needed. The process of stimulating respiration by adding ATP can go on until all the oxygen in the suspension of mitochondria is used up. When there's no oxygen left, there's no more respiration. By measuring the chart, the amount of oxygen which was taken up by the mitochondria when a known amount of ADP was added can be worked out. To show that ATP was produced at the same time needs a different technique. There are a number of ways of measuring the concentration of ATP in a solution. Some of them involve chemical analysis, but an alternative approach is to use a biological assay, and sometimes this is more sensitive than a chemical one. In a biological assay, the energy released from ATP when it's hydrolyzed by a biological system is converted to some other measurable form of energy. Glowworms and fireflies, for instance, have an enzyme system which converts the chemical energy of ATP into light energy. So, if desiccated firefly tails are added to a solution containing ATP, light is produced. Now, any ATP produced by these mitochondria when they were respiring wouldn't have stayed inside the mitochondria. It would have been transported out to the surrounding liquid, ready to supply the various ATP-using systems of the cell. So this suspension must be centrifuged to remove the mitochondria and get at the ATP. When the tube comes out of the centrifuge sometime later, it looks like this. The mitochondria have once again been packed into a cream-coloured pellet at the bottom of the tube, leaving a clear solution which should contain ATP. If this solution really does contain ATP, and it's added to this suspension of firefly tails, light will be produced. So if the two liquids are mixed, and the room light is put out, you can see the firefly tails glowing. What you are looking at is the conversion of the chemical energy of ATP directly into light energy, and the amount of light being emitted is proportional to the amount of ATP present. Using an instrument with a sensitive photomultiplier tube, ATP can be measured in extremely low concentrations, right down to 10 to the minus 12 molar. When the amount of ATP produced and the amount of oxygen taken up during respiration have both been measured, the two values can be combined to give a measure of oxidative phosphorylation, which is usually expressed as a PO ratio, that is, moles of ATP produced per atom of oxygen consumed. In the case of pyruvate, the nutrient added to the isolated mitochondria in this experiment, a PO ratio of 3 is usually found. In other words, when pyruvate is being oxidized, 3 moles of ATP are produced for each pair of electrons passed down the chain to oxygen. In these experiments, 2 things have been measured: oxygen consumption and ATP production. Although the results obtained tell us something about the process of oxidative phosphorylation, there's still a lot to be discovered. Many more years of research will be necessary before the intricate mechanisms of mitochondrial respiration are finally understood. All the energy for life on Earth comes from the sun. Green plants and certain bacteria use the energy in sunlight to turn molecules of water and carbon dioxide into starch and other compounds. This process is called photosynthesis, and in plants it takes place in the leaves and other green plants. Leaves are green because they contain the green pigment chlorophyll. It's the chlorophyll that turns the light energy of the sun into the chemical energy of starch during photosynthesis. This plant has been growing in the sunlight, so its leaves should have been photosynthesizing and making starch. Now, you probably already know that starch gives a blue-black color when it reacts with iodine. To show that this leaf does contain starch, all the chlorophyll is washed out of it with boiling ethanol. After a few minutes, the leaf is taken out of the ethanol, it's unwrapped, and iodine is dripped onto it. The blue-black color of the leaf shows that it does contain a lot of starch. A leaf that's been kept in the dark for 24 hours, so that it can't carry out photosynthesis, won't stain blue-black with iodine. Now, a solution of chlorophyll like this looks green because it reflects light from the green part of the spectrum. If it's put into a beam of white light, the light from the green part of the spectrum is transmitted, but the light from the red and blue parts of the spectrum is absorbed. But chlorophyll molecules that have been extracted from plants, like the chlorophyll in here, can't trap the energy of light they absorb. They emit it again as light of a different wavelength. A beam of blue light, for example, is absorbed by the free chlorophyll molecules, but then emitted in the red part of the spectrum. This process is called fluorescence. In order to trap the energy of light, the chlorophyll must be in its proper place inside the chloroplast. This is a chloroplast, magnified many times in an electron microscope. It's got an outer membrane, which separates the chloroplast from the cytoplasm, and a lot of internal membranes, which are either free or built into stacks. And it's inside the inner membranes that the chlorophyll molecules are arranged. The chloroplast contains everything that's needed to make starch. Not only the chlorophyll, but all the other enzymes and small molecules which are involved in photosynthesis as well. The large starch granule in this chloroplast is evidence that photosynthesis has taken place. It's quite easy to isolate chloroplasts from soft leaves like spinach. The leaves are put into a blender to break up the tissue. Then they're filtered to remove any large unwanted pieces of tissue, and the filtrate is centrifuged. You can keep track of the chloroplasts because of the green colour of the chlorophyll inside them, and after centrifuging you'll find that they've all been packed into a pellet at the bottom of the tube. By adding a suitable buffer solution and stirring, the chloroplasts can be resuspended, and when the suspension of chloroplasts is illuminated with the blue light, there's no fluorescence. Unlike the free chlorophyll molecules, chlorophyll held in its place in the chloroplast membrane can trap light, and this light energy can be used in making starch. Starch is a polymer built up from small carbohydrate units. During photosynthesis, what the chloroplasts do is to take carbon dioxide from the air and reduce it, using the hydrogen of water as the reducing agent, to form carbohydrate molecules, which are gradually built up into starch. The remaining part of the water, oxygen, is given off. Now, the process shown here, the reduction of carbon dioxide by water, is the overall process occurring during photosynthesis. The way photosynthesis takes place in the chloroplast is actually much more complicated than this, and takes place in two stages. In the first stage, light is absorbed by the chlorophyll molecules in the chloroplast, and electrons from water are transferred to a coenzyme, so changing it from its oxidized form, known as NADP+, to its reduced form, NADPH. At the same time, ATP is produced. This stage of photosynthesis is called the light reaction, because it requires the direct involvement of light. In the second stage of photosynthesis, the product of the light reaction, the reduced coenzyme NADPH, is used to reduce carbon dioxide to carbohydrate. This process also uses the ATP formed during the light reaction. The second stage of photosynthesis doesn't involve light directly, which is why it's called the dark reaction. But it's important to realize that in plants, this reaction doesn't normally go on in the dark, because the reduced coenzyme produced in the light reaction can't be stored for very long. Production of reducing power during the light reaction can be demonstrated by adding a blue dye to chloroplasts. The reducing power, which normally, remember, is used to reduce the coenzyme NADP, reduces the dye to a colourless form. And this change can be detected because, as it takes place, the chloroplasts return to their green colour. This type of reaction, where the chloroplasts reduce a substance other than the normal coenzyme, is called a Hill reaction. Now, the light reaction of photosynthesis is itself quite a complicated process, because the transfer of electrons from water to the coenzyme NADP requires the collaboration of two photosystems, or assemblies of chlorophyll molecules. Light collected by the chlorophyll molecules in one photosystem causes one special chlorophyll molecule to become excited and expel a negatively charged electron. This electron is transferred to the coenzyme NADP, and the place it occupied in the chlorophyll molecule in photosystem 1 is filled by an electron expelled from a special chlorophyll molecule in the second photosystem when its chlorophylls absorb light. The electrons lost from photosystem 2 are replaced by electrons produced by the splitting of water. The hydrogen ions from water are transferred to the coenzyme NADP to complete the chemical change from the oxidized form NADP+ to the reduced form NADPH. The oxygen is given off. And while all this is going on, some of the light energy collected by the chloroplast is also used to produce ATP. Once the coenzyme's been reduced, it can be used in the dark reaction to reduce carbon dioxide to carbohydrate. This process, converting carbon dioxide to carbohydrate, not only needs electrons, it also needs chemical energy in the form of ATP, and the ATP produced during the light reaction is used for this purpose. This equipment measures the rate of photosynthesis by measuring the rate of oxygen production. This is done using an oxygen electrode, which measures the concentration of oxygen in solution. The electrode itself is at the bottom of this chamber, and if the chamber is filled with a suspension of chloroplasts which are photosynthesizing, and therefore producing oxygen, the electrode can give a measure of the rate of photosynthesis by recording the increase in the oxygen concentration of the suspension over a period of time. A steep gradient on the chart recorder connected to the electrode shows that there's a rapid production of oxygen. A gradient that's not so steep shows that there's little oxygen being produced. The electrode chamber has been covered with silver paint to exclude all the external light, so that the chloroplasts only receive light of a known wavelength, which enters the suspension through these bundles of optical fibers. The fact that there are two photosystems involved in the light reaction of photosynthesis can be demonstrated because the two photosystems absorb light of different wavelengths. At the moment, the chloroplasts are receiving light with a wavelength of 700 nanometers. Now that's light from the far red region of the spectrum, and because they're green, the chloroplasts can absorb it very well. But they can't use light of this wavelength to oxidize water, so the output of oxygen at this wavelength is very slow. The gradient on the chart isn't very steep at all. But if light of a shorter wavelength is added, oxygen is produced rapidly, and the really important thing is that this production of oxygen is much faster than if the shorter wavelength alone is used. The dramatic drop in photosynthetic activity when the far red light alone is used, that's the light with a wavelength of 700 nanometers, is known as the red drop. What's happening is that the far red light alone excites only one of the two photosystems, the one involved in reducing the coenzyme, and this can't function without the other system. Because of this, The production of oxygen during photosynthesis requires the cooperation of photons of both far red and another wavelength. And the cooperation of both systems gives enough energy not only for the reduction of the coenzyme NADP, but also for the synthesis of ATP. ATP is needed together with the reduced NADP for the dark reaction. So, using the two photosystems together, the chloroplast can produce, in the light reaction, both of the intermediates needed for the dark reaction. The chlorophyll-containing membranes of the chloroplast are not needed for the dark reaction. They've done their work in producing NADPH and ATP. The dark reaction uses soluble enzymes of the chloroplast stroma. But that's another story.